our next presentation. This is on, thank you very much, Jorge, on physical geochemical modeling of the Gulf of Mexico by Julio Sheinbaum. I'm happy to introduce Julio. He's a specialist in numerical modeling, in physics and biochemistry, and in the collection, analysis, and assimilation of data for oceanic and meteorological forecasting, in particular of the Gulf of Mexico and the Caribbean Sea. Julio is a researcher level D at CICESE and level 3 in Mexico's National Research System. He completed his undergraduate and master studies in physics at the Faculty of Science at UNAM, and his doctorate at the University of Oxford in the United Kingdom. Since 1991, he has worked as a researcher in the Department of Physical Oceanography at CICESE in Ensenada, Baja California. He has been the coordinator of the work group on numerical modeling within the Gulf of Mexico Research Consortium, Sigon, that has been funded by the CENER CONACYT. Well, welcome, Julio, and we look forward to hearing and learning from your presentation.
Well, it's the same as Jorge. Thank you very much, Zenon, Jorge, and everybody here for inviting us to present a little bit of the work that we have carried out or did carry out for the Sigon project on the modeling side, besides the strong observational effort. And I'm going to talk a little bit about physical biochemical modeling, which originated with the idea that somehow we would sort of use these models in some way with the oil spill modeling, particularly with biodegradation processes. The truth is that in the end, we did the biodegradation process for the oil spill modeling in a different way, but we actually learned a lot from the physical biochemical modeling of the Gulf, and I will try to give you a small story of a lot of the work that we did. So if you go to the next one, please. So just a quick reminder, like Jorge mentioned, that the work that I will show you is actually based on the work on a lot of people. These are the names. I won't go through all of them. Just to mention that you know, it involved collaboration among different Mexican institutions, but also international institutions from several places. And so the collaboration, particularly with European institutions and US institutions, academic institutions, it's been going on for a while on the modeling and the observational side. The next one, please. So the main results that I'm going to show you were carried out particularly by Pierre Damien, who was a postdoc in our group. Now he's at UCLA working with Jean McWilliams, Sheila Estrada, who works, she's now a full research scientist at CICESE, and several other people, Orenz, who did some, I'm going to show some observational work that he did, Julien Joanneau from Lagos in France, Olaf Duteil from Geomar, and Joao Sousa now at the New Zealand Met Office. The next one, please. So I will try to give you the, you know, kind of a short view or a summary view of the scientific story that we are facing in the Gulf of Mexico and that we learned from several observations. So this is pictures from the analysis of a very nice paper by Benjamin Martinez Lopez and Jorge Zavala, where they analyze the chlorophyll observations, satellite observations in a very nice way, and produce these maps which show very clearly the seasonal cycle at the surface of the chlorophyll. And obviously, what you see on the left panels is winter and fall. And on the right panels, beginning on the first one, May, so you have the end of spring and summer. And obviously, you see the strong variability in terms of the chlorophyll content at the surface, right? And so we know that the Gulf of Mexico is an oligotrophic system, and particularly in the summer, when you have a very stratified waters and warm waters, the signal of the chlorophyll is really very weak. You see, obviously, the impact of the Mississippi River and some of the rivers. You see also something that I will talk a little bit at the end, some of the, or part of the signal of the upwelling in the Yucatan Shelf, right at the north of the Yucatan Peninsula. And on the left-hand side, you also see these, in some of these places, you see these jets that emerge from the coast, particularly in the, one in the border between the US and Tamaulipas, Texas and Tamaulipas, and one in the south near the western part of the Yucatan Peninsula. I don't have the mouse, so I cannot show you. But anyway, so the clear seasonal signal at the surface usually is translated or extrapolated to the deeper ocean, at least the near surface ocean. But is this really the whole story? If you go to the next one, please. So beginning with the observations from the Sigon project network, this is on the left-hand side, you see the stations that probably, if somebody hasn't talked about them, they will in the near future. So this is from Sharon Herska, who provided this this picture with all the Sigon stations in the southern Gulf of Mexico. And besides that, during 2011, well, actually from 2011, 2017, more or less, there was a big project funded by BOEM in which several biogeochemistry floats were deployed in the Gulf of Mexico for 5 years. And you can read a lot about this project in the paper by Hamilton, but there is also a famous paper by my colleague Paula Perez Brunius, who is speaking after me. But the biogeochemistry data, or the biogeochemistry floats, provided unprecedented information about the fluor, or chlorophyll content, and a lot of the useful biochemical information below the surface. And if you go to the next one, please. And this work, which was guided by Orenz, he has a long name, Orenz Pasqueron de Pomerbeau. If I actually, I guess I didn't pronounce it properly, but anyway. So he was a postdoc also working with Paula and with us at CICESE for a couple of years. And he took the task of analyzing all these data from the biogeochemistry floats. And here in the top panel, you see data from different floats, seven or six, well, actually 7 different floats, which at the top you see, in the x-axis, the horizontal axis, you have time, and then the top one is the mixed layer depth. The one in the middle is, if I'm not mistaken, is the surface chlorophyll measurements, and then the one at the bottom is the total chlorophyll content, right? So is the chlorophyll integrated from 350 meters more or less to the surface? And what you see particularly here, I mean, it's pretty evident in the top 2 panels that you have a strong seasonal cycle with the deep mixed layers depths during during winter, and also you have the strong also seasonal signal with a high chlorophyll surface content at well, near the surface chlorophyll content during winter, and very low chlorophyll surface content in the summer. But then the one at the bottom, with all, you know, all the floats, all these Villargo floats, show that the actual integrated chlorophyll content in the water column has a very, or I would say, relatively or even absent. There is an absence of a clear seasonal cycle. So this was very surprising for us. The next one, please. So to make the story short, something that we found with these Villargo floats, and which was very challenging to try to understand, was the idea of, well, what is happening? I mean, why this is not, the seasonal cycle is not showing in the whole chlorophyll content in the water column, only at the surface. And so we came up with an explanation, which we actually divided between cyclones and anticyclones in the Gulf of Mexico, and essentially suggesting that what happens is that during winter, particularly during winter, when the mixed layer goes deep, it doesn't actually goes as deep as the nutricline. So even though there is some deepening and obviously an extension, a deep extension of the mixed layer doesn't reach the nutricline, and it's unable in general to introduce new nutrients into the euphotic zone. And so whenever you see high chlorophyll concentrations during winter, it's not necessarily that you have new primary production, that you have new production, but that you are actually mixing the surface chlorophyll max, the subsurface chlorophyll maximum that is shown in these green bumps that you see over there. And the signal is a little bit different in the cyclones and the anticyclones. And one of the things that you see particularly is that the mixed layer is deeper in the anticyclones, and this is something that I will talk about later. But the main message here, it's already that I want to, you know, for you to go, or the take-home message, if you like, is that what you see at the surface in terms of chlorophyll is not really what you see in the whole water column, and this is something that needs to be explained. The next one, please. So getting into the numerical models, we have two numerical models, one which was mainly carried or developed by Pierre Damien, as I was telling you, and this is the NEMO-PISCES model. The biochemical model is called PISCES, and it means Pelagic Interaction Scheme for Carbon and Ecosystem Studies. It's a very complicated biochemical model, 24 compartments with 2 phytoplankton compartments, the small and large phytoplankton, the same for zooplankton, and several nutrients. I won't go into the details. And the hydrodynamic model, or the physical model, is the NEMO model. This is the nucleus for the European modeling of the ocean, and it's a model that we have been using at CICESE for a while. And as you can see, this model has about 8-kilometer resolution, 75 levels in the vertical, and it was spun up for 75 years to do the analysis that I'm going to show you. The idea with the model, if you go to the next one, please. Well, first was to try to see if we could reproduce a little bit, or more or less, the observations in the Gulf. So we did a lot of tuning, and the work is, you can look at the work in this first paper that came out of the analysis. And what you see in these pictures is the distribution of chlorophyll in the vertical. So the z-axis, or the vertical axis, is depth, and the red curves are the model, and the gray curves and the gray dots are the observations. And the model as usual, is not perfect, but we claim that given the limitations of these models, that the model is actually doing relatively well. If you go to the next one, please. So after tuning the model, we said, well, can we explain a little bit of, or even go into a bit more detail about what is happening with the distribution of chlorophyll in the surface and in the integrated or the fully integrated content in the water column. And some of the results I came out from this model are that you can actually divide the Gulf of Mexico in 3 different regions based on how deep the mixed layer goes and how does it interact with the nutricline. And so the first region, you see here two sets of plots. The one at the top is the surface chlorophyll. The one at the bottom is the integrated chlorophyll in the water column, or the fully integrated water chlorophyll content in the water column. And you see the first region, which is the Bay of Campeche and part of the eastern Gulf of Mexico, that there is a seasonal signal, a weak but a seasonal signal in the surface chlorophyll, well, the surface chlorophyll produced by the model, whereas the integrated chlorophyll is really flat. There is no variation during the year. The x-axis is month during, is the climatology. So this is month, number of months, right? From 0 or 1 to 12. So then you have the one in the middle, where you have the situation where the signal is, it's a mixed signal where you have very clear signal, seasonal signal in the surface chlorophyll, and also a relatively weak signal in the total chlorophyll content. And then you have the northern part where there is a clear seasonal signal both in the surface and in the total content of chlorophyll. And what explains this is the idea that in these regions, or these different regions, the connection between the mixed layer and the nutricline is determining the presence or absence of this seasonal cycle. The next one, please. So these are the 3 regions, and as I was saying, so in the northern part you have the region where there is a clear increase in biomass. There is no such a clear increase in biomass in the first region, in the Bay of Campeche, and the middle one, where the eddies are usually found, is kind of in between. The next one, please. So I will go very quickly through this, but the idea is, okay, we now look at the relation between the mixed layer depth, the relation with the nutricline, and whether there are regions where there is new production of new biomass production, or there is no real new input of nutrients into the euphotic zone. And the question is, how is this connected by the presence of eddies? So I will have to, please, if you go to the next one. I don't have much time, so just to tell you very quickly, this is again a climatology. The red ones are the cyclones, the blue lines are the anticyclones, and you are seeing again the surface chlorophyll content and the total chlorophyll content. And again, the cyclones tend to be a bit have a little bit more content in chlorophyll, but it's obvious that the seasonal cycle in the total chlorophyll content is smaller than at the surface. And one of the things that we would like to understand is what is happening, particularly with the anticyclones. So if you go to the next one, please. And one of the things that we found is very interestingly is that whereas you can see a negative anomaly, I mean, if you analyze, you follow the eddies, you try to follow the eddies in the model, and you look at the surface chlorophyll content both in well, surface and total chlorophyll content, and you look at the anomalies, what you see is this strong negative anomaly of this chlorophyll in the total content of chlorophyll in the summer in the anticyclones, but a positive anomaly during winter in the anticyclones. The next one, please. So, you know, to try to make the story short and quick, the idea is that, again, you know, the take-home message, there is a difference between the total chlorophyll content and what you see at the surface. What happens, or what we believe is happening, is that because the anticyclones are less stratified, it takes less energy, or the same energy that is, for example, put by the winds, or when you have a very strong cold front going through the Gulf of Mexico and interacting with these eddies, that on the anticyclones, the energy required to do the mixing is sufficient to make the mixed layer, which is deeper in the anticyclones, interact with the nutricline and bring nutrients to the euphotic zone and increase the chlorophyll content due to new production. This is the figure on the left that you see there. The bottom panels show that in the anticyclones, you actually have, based on the model calculations, obviously, you have the new production is higher during winter in the anticyclones than in the regenerated new production. And this is, we are explaining that based on this interaction between the mixed layer and the nutricline inside the anticyclones. The next one, please. And changing the subject, this is something that we did with this Nemo-Pisces model. We use the ROMS model now to analyze another very interesting problem in the Gulf of Mexico, which is the upwelling system in the Yucatan shelf. The panels on the left show the chlorophyll signal at different seasons, and you see particularly that during well, this is in the model actually, but so during winter and spring, you see that there is a seasonal signal, and the bottom panels show a cross section along this along the Yucatan well, peninsula, if you like, at about 21 or 20, yes, 21 north, more or less. And so in the these two panels, you see the signal in spring and in autumn, and you can actually see the upwelling of cold waters going into the shelf coming from the Yucatan Channel. And so this work made by Sheila Estrada and all the collaborators actually looked at the nitrogen balance in the Yucatan shelf and in the whole area, and show, if you go to the next one, please, show that the Yucatan shelf is exporting nitrogen through the north and through the west through several processes, on the west involving coastally trapped waves, and it's getting recharged, if you like, of nitrogen by the underground water sources, but also from the upwelling in the Yucatan shelf, which is not, the important thing here is that this upwelling is not produced by the winds, it's produced by the interaction between the Yucatan current and the topography of the Yucatan Peninsula. And details can be seen in this paper. Yes, I
Julio,
know I have to finish.
yeah, Julio, I'm sorry to interrupt. How far you go?
No, that's the last one.
Very good.
So if you just go to the next one, please. So let me quickly just say that we are embarked now in another project where we do a very high resolution measurements and modeling of one of these loop current eddies. You know, taking a ship and making measurements and doing modeling. Won't go into the next into the rest of the things. And if you go to the next one, you can see a lot of these the comparison. The next one, please. No, there should be another one. Oh no, I'm sorry, you don't have that one. Well, no, I made a modification to mine. It's just that at the modelacion.sigon.org, I will put it in the chat. You can see a lot of the different results from the different models. And I'm sorry for taking a bit more time. That's it.
Thank you, Julio. Because of the time,
yeah, go ahead.
I would suggest that we move to the next presentation. And if you have any question, please formulate it on the chat box, and we're going to pass it to Julio, and I'll ask Julio to respond via email or by arranging a meeting with him later, maybe. Our next